Hello everyone. Today we will see endospore staining. First, what is mean by spore? Spores are the resistant forms of bacteria. There is another form called as vegetative form. Spore is the resistant form that can withstand even higher temperatures and that can be killed only by autoclaves at 121 degrees Celsius. So, is that important to stain a spore, or is it mandatory to observe it under microscope? Yes, we should know whether the organism is sporulating or non-sporulating is one of the characteristic feature of a the bacteria. So it is of course important to see the spores under microscope. When we want to see the spore, it's better to view under phase contrast microscope, unstained wet films. So where the spores appear refractile, oval or spherical bodies within the bacterial mother cell, or else it will be free from bacteria. So we require a phase contrast microscope in that case for the unstained wet films. If we want to stain them, we need some kind of endospore staining. They are very weakly acid fast. So I would have explained you what is acid fastness under acid fast staining category. I'll give the link in the description. These spores are very weakly acid fast. So similar to the acid fast staining, we can apply heat and we can make the spores to get stained, and we can have a counter staining for the background. That can happen. So if spore bearing organisms are stained with ordinary dyes like gram staining, the body of the bacillus alone will be deeply colored, whereas the spore is unstained. It will be appearing like a clear area in the organism. This is the way usually the spores commonly observed. But if we desire, we can make a vigorous staining procedures to introduce the dye into the substance of the spores. For that, we can make a modified Ziel-Nielsen method with 0.25% of sulfuric acid, or malachite green stain can be adapted. Usually malachite green stain is adapted. So what are the bacteria that produces spores? We can broadly classify the bacterium into aerobic spore bearing, anaerobic spore bearers, likewise aerobic non-spore bearers and anaerobic non-spore bearers. Aerobic spore bearing organism example is Bacillus anthracis. Aerobic non-sporulating organism example is Escherichia coli, Klebsiella pneumoniae, Salmonella, Shigella species. All these are non-sporulating. They are facultative anaerobe too. When we come to the anaerobic sporulating organism, Clostridium difficile, Clostridium perfringens, Clostridium tetani, Clostridium botulinum, all these are sporulating. Non-sporulating anaerobic bacteria are, for example, Lactobacillus, Veillonella, Bifidobacterium, all these are non-sporulating anaerobic bacteria. When we see the procedure for this malachite green stain for spores, this is actually called as the Scaeffer-Fulton method, where we have the modification by Ashby also. Here the films are dried and fixed with the minimal flaming. The smear, in the sense the films, to make a smear we will take the suspected colony, that has been sporulating colony. So with the smear we have to make a thin film that has to be dried and fixed with the minimal flaming. We have to place the slide over a beaker of boiling water, resting it on the rim with the bacterial film uppermost. So to apply the heat, we are not supposed to give a direct heat. Instead, we have to place it on the boiling water, so that heat itself is sufficient to take up the stain by the spores. When within several seconds, large droplets have condensed on the underside of the slide, so that heat itself is enough. Flood it with 5% aqueous solution of malachite green and leave to act for 1 minute while the water continues to boil. Now wash the smear with cold water. Treat it with a 0.5% safranin or 0.05% of basic fission for 30 seconds. Then wash it and dry it. If you observe the smear now under oil immersion, our spores will appear malachite green color, the green in color, because the primary stain would have been taken by these weakly acid fast spores, and the background, the bacilli, will be red in color. There is one advantage of this malachite green stain, that is, if you do the modified Ziel-Nielsen technique, even the lipid granules will get that pink color or that red color, whereas in malachite green we can avoid that lipid granules to take up the stain. That is the advantage over performing modified Zeal-Nielsen for spore staining. It's better to adapt to the malachite green stain so that we will see the true positive spores. Granules will not take up the stain in malachite green staining, that is Scaeffer Fulton modified staining should be adapted for endospore staining instead of modified Zeal-Nielsen staining with 0.25% sulfuric acid. Hope you have understood. If you have any doubts, you can make it in a comment section.